Welcome to biotechnology. Today we're interested in a thing called an operon. This is how cells turn their genetic information on and off. An operon works a little bit like a piece of computer code. So if we imagine we want a robot to drive around and do something, we would like it to not run somebody over. So it will have a sensor on it that is looking all the time to see if there's somebody there, and if there's somebody there it will do something, its emergency stop or something like that. That is very much like an operon works, but it works in a chemical manner. So if we were going to write that into computer code, we would have a little piece of subroutine, a piece of code that says watch, watch, watch on the sensor. Is there a squishy person nearby? And when it received a reply from that sensor, it would turn everything off, and that would be running in the background while it was doing everything else. That's very much like an operon. So the operon is a piece of genetic code that contains some instructions of what to do when the event happens that it's looking for, and it has some switches that turn it on or off in response to events. The most common operon that we're interested in biology is the lac operon, because it's been very well studied and it's often misused. It's often been altered by biotechnology to be used by us. So if we draw the lac operon, for example, we will have some way of turning it on and off, and then all of the codes that it's going to do, all of the things that it's going to do afterwards. Let me draw that. So just like a computer program, we have a start to our code, and then it goes through effectively yes-no set of questions. Is there glucose present in the environment? Because this is a piece of code that makes lactase and some other proteins related to it. So it's a protein that a fungus would have, or a bacterium would have, if it needs to digest lactose. And if it wants to digest lactose, there is no point doing that if there's glucose in the system. So it would be evolutionarily useless for it to make lactase. If there was glucose there, it's much better eating that easy food. So it does that instead. It's lazy. So if there is glucose there, it doesn't make lactose. It also needs to check if there's any lactose in there, because there's no point making lactase if there's no lactose there. So it checks to see there's no glucose and that there is lactose, and then it starts making the proteins. We could draw that like a computer code. So here it is drawn out more like a computer diagram. So we would have start the program, then we'd have some kind of defined wait, because if it's a computer program, you have to define the amount of time you wait for. In this case, it's defined by chemistry. Is glucose present? If it's present, then it goes back to the start and waits again. If it's not present, it goes to the next one, checks to see if lactose is present. If lactose isn't present, it goes back to the start. If it is present, it makes lactase. And it also makes all of the other things that are on there, which in this case are molecules that help the lactose get through the cell membrane, and other types of things that carry on once the degradation has happened, once it's been broken into parts. So they're all connected together. This can be one protein, obviously, but it can also be more than one, and in the natural form of the lactase operon, there are several. But these are not computer programs, they're chemicals. So how are these going to work? We're going to need some kind of molecule that when it sees glucose on it, it attaches to the RNA. So let's think about that right now. Okay, I've confused myself now, so I'm going to have to start again. So this is our DNA. This is our DNA, and we need to copy the DNA into RNA. So we've got DNA, RS. So this will be a little bit weird with time for you, but let me try it again. This is DNA, so we start with our information on DNA. We need to make it into RNA, because it's RNA that is copied into proteins. It's really the sort of working copy of the genetic information. So DNA gets converted into RNA, and the RNA gets converted into protein. But it's not really converted, it's transcribed, meaning the information is copied over, but it's converted effectively into a different language, the language of DNA, into the language of RNA, into the language of a protein, and the original copy is not destroyed. So our DNA can make as many RNA molecules as it likes, and the RNA can make as many protein molecules as it likes, although obviously they do get broken down over time. So the DNA is transcribed, it's copied into RNA, which is just single-stranded, and it's the DNA to RNA copying procedure where these switches occur that you're about to see, because the switches act to prevent the DNA being copied. So on our start for the DNA, it's a promoter. The start of the DNA is a promoter, and it copies a section of RNA, and the start for a protein is a start codon, which is 3 bases of RNA, and that causes a protein to be made from just this section after the start, between the start and the end. There is other information on this RNA. In a bacterium there can be several. Start, end. In a bacterium there can be several proteins coded on the same strand. In animals and us, it's typically not. It's typically just one protein for one strand of RNA, but it's just a difference between species. The main thing that I may have been confusing about, and I will have trouble cutting this to make it work, is that the promoter and these genetic switches work on making RNA, on controlling how much RNA is generated, and the RNA is automatically converted into protein. There may be other ways of controlling how that works, but once we've made RNA, it will start to make protein on its own. So just to make it full, we've got to first of all work out what's going on here. So the DNA in our cells, in our genetic material, is converted into RNA. We're going to be thinking here mostly about bacteria because they're more simple, because in bacteria the whole code for protein is relatively clearly copied directly from DNA to RNA, and not very much happens in between. In higher animals, little bits get cut out. They're called the exons and introns, and it all gets put back together again and becomes the information to make a protein. But in bacteria it's a little bit more simple. It's a direct copy of the DNA. In converting this RNA data into a protein, there isn't enough information in 4 bases to make 20 amino acids, to code for 20 unique amino acids, and therefore we need to use sets of them, or biology needs to use sets of them, and it uses sets of 3, which gives it a lot more codes than there are amino acids, but that's alright. It's a lot better that way around than the other way around. So we've got sets of 3 RNA bases coding for 1 amino acid. But at the start we need a little section that's going to stop, it's going to block it when there's a molecule on here that's attached to glucose. Let's have a square for glucose. That's our square for glucose, and it's going to attach to a few of these bases on the RNA and prevent the copying machinery from making a protein. So that's all right. That would be our glucose. What about lactose? It's much the same. So if it's much the same, it means this molecule attaches if there isn't any lactose on it. So let's have lactose being a red triangle. If the red triangle comes along, the shape of this molecule changes. It changes configuration. That's not visible to you, is it? The shape of this molecule. Okay, so I'm back. Where's my pencil? So the shape of this molecule changes and it lets go. This would be checking for lactose. So red, lactose. And this one changes shape. Let's draw it as this little cleft closing when there's no glucose in there. It's not obviously exactly that shape. It's different, but it's a good way of drawing it. So it changes so it can't attach to the RNA. This one changes when it's attached to the lactose. This one changes when it's not attached to glucose, and that allows us to have the correct sign for our two questions that we want to ask. But it does generate a problem, because this won't work unless we can make these two molecules, and they have to be made somehow. So we have to have some code for them somewhere along our genetic material. Where is it? If it's behind the start, it can't be made until it gets past it, and so we will always make some lactose before being able to block it. So in a more realistic example, we might want to get bacteria to make spider silk. Spider silk's a very effective material for implants. It's relatively body friendly, so you can put it into somebody's body. It will get broken down, but very slowly, so you can use it to replace parts of people's bodies, and it is gradually replaced by normal material. It takes quite a long time, which is good, because then it can do its job, its material job of holding things together before the body takes over. So we might want to make spider silk, and it's very expensive to make if we extract it from spiders, not because spiders are difficult to milk their silk, but more because spiders are expensive to keep, because you have to feed them on something that would be live insects, and that's obviously difficult. So it costs more money to make spider silk than you would imagine, or maybe you can imagine exactly how much money it costs. So we might want to make it in a different way. We might want to get some bacteria to make it for us, and so we could replace this section where the protein, where the lactose was, with the spider silk code from a spider. It's not quite as easy as that, but first of all we will look at the problem that we can solve with this. So what is the problem? Spider silk isn't very soluble, so when the cell makes it, the spider silk will clump together and drop out and make a blob inside the cell and eventually kill it. That's obviously really bad. If we spend a lot of money making some cells, converting them into special cells that have this code, and then they make a little bit of silk, just a tiny bit each, and then die, that's not very useful. What we want to do is to make them multiply a lot, and then we can sacrifice them. Then they can all make a little bit of silk, because the advantage of bacteria is they're cheap. Inside a tiny volume there are millions of bacteria, so it's easy to make a lot. If we have a few bacteria, even just one, we can make millions within a short time. So effectively for E. coli, the bacteria that people often study, the generation time is about 20 minutes, so every 20 minutes the population doubles, and that means we can get up to huge numbers within a few days. So typical bacterial swab, if we take a petri dish and we put one bacterium on it, we'll be able to see the colony in a day or two. So the obvious solution to this problem, that the silk kills the cell, is to not make it until we've grown a few cells. So we would like to turn the silk off. And we can do that with one of these two switches, so we can abuse them to do the things that we want. The lactose switch is slightly more helpful. So if we don't worry about the glucose one, so we break that so it doesn't work, and we take the lactose one, then if we grow our bacteria in a medium that contains no lactose, we can make no spider silk. We can make all of our cells divide and make lots and lots of cells, and then we can switch it back on again by adding some lactose. There's a problem though. If we add lactose, this process will turn on, and they'll have a normal one as well that will break down the lactose, and then it will stop making silk as well. So that's not ideal. Ideally, we would have something that tricks this, that switches this switch on and causes it to make our spider silk without having to put lactose in there. There are two normal variations to this. So in industry, one of the variations is you really do go from glucose to lactose. So we really do grow our bacteria in a solution containing lots of glucose, and then we add some lactose and allow the bacteria to eat all of the glucose, so it switches effectively the same as it would have done in the wild, but instead of turning on lactase, it turns on whatever thing we want to make, possibly killing the cells, but making an awful lot of the protein that we want. So that's a comparatively simple way, although every time we do it, we have to control the conditions very carefully so that we make the things that we want, because this switch isn't exactly that easy. So the alternative is that we take one of these switches where there isn't a normal form, where the bacterium that we've chosen, in this case, can't break down lactose at all. It doesn't have a natural lactase gene set. And then we can add lactose, and because we've broken this one, it can't break down the lactose. This is the case in our biorad experiment. So it's an arabinose digesting switch, and in the presence of arabinose it turns on our protein, but because the bacteria that we use can't digest arabinose, then we can add a little bit and it never goes away. It just stays there the whole time, holding this switch on the whole time, very much like if we had a computer program or an electronic set and we've fixed the switch on, then it's broken and we will make spider silk and nothing else in that case. So if we look at our RNA, we have 3 bases, which are our codon, and there are only 4 possible bases in RNA. So we have one of 4 bases 3 times, which is 3 to the power of 4 possible combinations, which is 64, 4 times 4 times 4. And we have 20 amino acids, but we also have, of course, the start codon and the end codon, and we have a few other things, the origin on the cyclic DNA and a few other things that we'd like to use, so we don't have quite so many extra ones, but we have about 3 possibilities per amino acid that we could use. This turns out to be the case in nature. So instead of using just 20 single unique codes for each amino acid and leaving all of the other ones for later work, what they actually do, or what we actually do, what organisms do, is there's a general layer of variation of possibilities. So one amino acid can have several different codes, very often 3, but sometimes 2 or sometimes 4. So it will often have several possibilities that we can make the same amino acid. That gives us a possibility, however, to steer how fast we make that, because these bases on here, this codon, is going to be attaching to another piece of RNA. Oops, let's put RNA on there. It's not very good because they aren't the actual letters. It's going to be attaching to a piece of transcription RNA, which is going to attaching to a piece of RNA that brings along an amino acid with it, And obviously there are, with this same amino acid, so let's call this lysine, one of the possible amino acids. If we have some lysine, we would be able to use various codes here to attach to it. But in each organism, there is a preferred code for one amino acid, one that is more common inside the body. So there are a lot of this molecule to ferry the lysine to our RNA to make the protein. If we have one that's uncommon, it will get stuck here and have to wait until one happens along. And that is one way where we can vary the amount of different proteins that are made, even though they're on the same strand. In the bacterium, they're on the same piece of RNA. It doesn't actually stop in between these, so we've got one piece of RNA with all three proteins on, and we might want to make more of one than another. And an easy way to do that is to choose codons that are rare, so that for the proteins that we don't want very much of, and codons that are common for the proteins that we want more of. In every case, we will have to tailor this to an individual. This can be a problem if we're doing our biotechnology. So in our case, we're converting the green fluorescent protein code from one bacterium to another one, and it could be that it wouldn't be very effective at making it because it uses common codons in the source organism, but they're rare codons in the target organism. There are not very many of them in there, and then we won't be able to make very much. So one of the tasks of the biotechnologist is sometimes to alter this code, to edit it slightly, to make it work better, to make it have more common codons in the target organism, so we can make more of the stuff that we want to make, in our case green fluorescent protein. The good thing about green fluorescent protein is it's a very small molecule, so unlike the silk, it is not toxic. It doesn't interfere with the process at all. It doesn't cost it very much energy, and we don't need to make very much because it's fluorescent. It's relatively easy to see. We don't need to make very much, and so it doesn't take over the bacterium and stop it from doing its normal stuff. It doesn't use up all of its energy. So to sum up, and to get it right this time, DNA, a signal on DNA, the information on DNA is copied from a promoter to the end of the signal, but there are other molecules that can block it and control how much RNA is made from one piece of DNA, and those switches are very important and really useful for biotechnology because we can use them to ask the bacterium, in effect, to make our molecule, whatever we want, at the time that we want it and not before. Very often that's because it's toxic. If we do that, we can make our target molecule and we can extract it more easily. If we make them use all of their energy to make a protein, that's good for us because then they've got more of the protein. It's a lot easier to get it back out again, because as we saw in our extracting DNA, most of the material inside the cell is not the things that we want. It's always the case. Most of it's water, and then there are a lot of other things. There are a lot of proteins, there are a lot of other things in there that we don't want. Purification of the product is one of the most expensive steps in our biotechnology set. So now we can make RNA, and the RNA uses a different code to make proteins, and there is a switch on the DNA to control how much RNA is made, but there's also a throttle on the RNA that controls how much protein is made, which is how common the codon is for the amino acids that are on the set, on the protein that we're using. If they're really common, it will make it faster in that organism, but the organisms use the same code for the same amino acid, but they don't have the same amino acid codons that they prefer. So if we steal a gene for something from one organism and put it into another one, it might be made faster, it might be made slower, it might be held up by one amino acid that's different, that's not common in that other organism. And we just need to fix that. That means editing molecules. So not so easy. There's a degree of redundance. That's how it can happen, because the same amino acids, there are 3 times as many codes as there are amino acids. So we have plenty of extra signals in order to carry this information of effectively how much of the protein compared to the other ones the organism wants. Why do we want these proteins in a package? Well, naturally it would be a response to something. So we would have proteins that all do the same thing together. They have a job together, and so they're related to each other. In the case of the lac operon, it's lactase, which breaks down lactose. It's a chaperone molecule that gets it through the cell membrane from outside.